Hello, this presentation focuses on how cells communicate with one another. It is intended for students taking Anatomy and Physiology 1 at Madison College with Gina Piscatelli. This field of biology is often referred to as cell signaling, with the idea that the communication is a signal that one cell sends to another. In this presentation, I will cover three main topics of cell signaling. First, the purpose, or why cells communicate with one another. Secondly, the two general methods by which cells do communicate. And lastly, the specific mechanisms involved in chemical communication, known as chemical cell signaling. It might surprise you to learn that cells communicate with one another, but they need to do so in order for the body to adapt to changing internal and external conditions. So let's first look at how cells might respond to a signal. Depending on the type of signal and which cell receives that signal, there are a variety of ways in which a cell might respond. A couple of examples include by dividing and by contracting. In the first example, division, suppose a damaged skin cell releases a chemical before it dies. This signal tells neighboring cells to divide. Therefore, dying cells are replaced with new cells to keep a tissue healthy. The second example, contraction, involves a neural signaling cell which directs the receiving cell, a muscle cell, to contract. Another common way a cell may respond to a signal would be to start the process of transcription, thereby ultimately producing a protein. The picture shows the signal in pink. The receiving cell is blue and has a purple receptor for the signal. When the signal binds to the cell's receptor, the process of transcription is initiated and the gene is expressed, or a protein will be made for that gene. Now that you are aware of some reasons why cells communicate, let's move on to the second part, methods of cell-cell communication. There are two general methods that cells use to communicate with one another. The first method is called direct contact, and the second method is called chemical signaling. In direct contact, the signaling cell and the recipient of the signal, or the target cell, are close to one another. In fact, they are bound together. The signaling cell has a protein signal embedded in its cell membrane, which binds to a receptor protein in the membrane of the target cell. Notice that in the picture of direct contact on the slide, the signal is drawn as pink and the receptor is drawn as green. Receptors are commonly drawn in this manner to show that they have a specific site where a signal binds. Also note the term ligand written on the slide. Any molecule that binds to a receptor is commonly referred to as a ligand. Therefore, signals used in cell communication are also called ligands. Now, the second method is often referred to as cell signaling, but more accurately it is called chemical cell signaling or signal transduction. I will use chemical cell signaling. This method also involves a ligand and a receptor interacting, but in this case the signal is not embedded in the signaling cell's membrane. Instead, it is released by the signaling cell. When the chemical signal is released, it must reach its target cell by the process of diffusion. Cells release many chemical signals that travel throughout the bloodstream as well as throughout the interstitial fluid. And of course, a cell is exposed to many chemical signals. but only cells that have a receptor for that specific signal will respond. A cell does not respond to all signals. This concept is referred to as specificity. The ligand and receptor are specific to one another, and a cell can only respond to a signal if it has a receptor specific for that signal. While it is true that a cell can only respond to a ligand if it has the specific receptor for that ligand, it is also true that a cell may be able to respond to multiple types of signals. This occurs when a cell has many receptors. In the example shown on this slide, the cell on the left, yellow, can only respond to hormone A because it has a receptor for hormone A. But the cell in the middle can respond to both hormone A and hormone B because it has two different receptors, each receptor specific for one hormone. In this last part of cell signaling, we will focus on chemical cell signaling, and there are 4 categories. These 4 categories involve a signal secreted by a cell, but how far the signal travels and the type of target cells differ. The 4 categories are paracrine, autocrine, endocrine, and synaptic signaling. 2 of the 4 types are shown on this slide. Paracrine signaling, shown in the top right of the slide, involves 2 cells that are close to one another. The signaling cell secretes a ligand that diffuses a short distance to reach the target cell with receptors for that particular ligand. Because the ligand moves by diffusion from a region of high concentration to low concentration, the strength of the signal would diminish with distance. Therefore, nearby cells are targeted. Autocrine signaling, shown in the bottom left of the slide, involves a cell which acts as both the signaling cell and the target cell. In this case, a cell responds to its own secreted signal. This occurs when certain immune cells are activated via foreign pathogen, which causes them to divide. You will learn more about it in A&P 2. The 3rd and 4th types of chemical cell signaling are depicted on this slide. Endocrine signaling, shown on the right, involves a signaling cell that's typically located in a gland. This cell releases a signal called a hormone. Because the target cell is far away from the signaling cell, the hormone must travel through the bloodstream to reach its target cell. When the hormone is released, it diffuses towards the bloodstream, but the majority of the distance it travels is through blood flow. The last type is called synaptic signaling and is shown on the left. Its name is derived from the fact that a neuron is always the signaling cell, and the junction between a neuron and its target cell is called a synapse. At the end of the neuron, a ligand, known as a neurotransmitter, is secreted or released. The neurotransmitter diffuses across what is known as the synaptic cleft to reach its target cell. The target cell may be a muscle cell, another neuron, or even a glandular cell. Because the signaling cell and the target cell are close to one another, this is just a specialized type of paracrine signaling. Now we are going to look more closely at the molecular level of signals and receptors. There's 2 main types of signals or ligands and 2 main types of receptors. These types may be used in any of our 4 general methods of chemical cell signaling: paracrine, autocrine, synaptic, or endocrine. It will become most relevant to you when you study the nervous system and the endocrine system. The 2 types of ligands are based on whether they like water or not, meaning whether the compound is polar and hydrophilic or nonpolar and hydrophobic. From your study of organic molecules, you may remember that any compound that is lipid or steroid-based is considered hydrophobic, but all the other types of compounds, proteins, carbohydrates, and nucleic acids that you learned about, those are all hydrophilic. Generally, ligands are either lipids or proteins. The two types of receptors are both proteins, but located in different regions of a target cell. When the protein receptor is embedded in the cell's membrane, it is called a membrane receptor or a cell surface receptor. When the protein receptor is located inside the cell, either in the cell's cytoplasm or nucleus, it is called an intracellular receptor. For a ligand to reach an intracellular receptor, it must be able to cross the cell membrane. Therefore, only hydrophobic ligands, a lipid or steroid, bind to an intracellular receptor. Ligands that are hydrophilic do not cross the membrane, and they will bind to cell surface receptors. Now we'll look at what happens when a ligand actually binds to its receptor. Let's first start with intracellular receptors. The mechanism involves a ligand that is hydrophobic, which crosses the cell membrane. Some examples include cortisol, testosterone, estrogen, and vitamin D. The receptor for any of these may be in the cytoplasm or the nucleus. When the ligand binds to the intracellular receptor, the most common effect is to regulate gene expression in the target cell. This means that a gene will either be turned on or turned off. If a gene is turned on, that means it will be transcribed and the protein it codes for will be made. If a gene is turned off, it means that the gene will not be transcribed and therefore the protein it codes for will not be made. This is how steroid hormones work. Looking at cortisol as a specific example of a hydrophobic ligand that binds to an intracellular receptor, we can see that the steroid hormone, or cortisol, actually can go through the cell membrane, shown here as the triangular molecule, and go through a nuclear pore to reach the intracellular receptor. Together, the receptor and cortisol bind to DNA. This promotes transcription of a gene that codes for an enzyme. The function of the enzyme is to catalyze lipid and protein breakdown. Now the cell can perform this function and use lipids or proteins for ATP production. Now we'll look at the mechanisms and effects that occur when a ligand binds to a cell membrane receptor. Remember, the ligand that binds to a cell membrane receptor is hydrophilic. It does not cross the cell membrane. Some examples include insulin, epinephrine, and acetylcholine. The actions of ligand cell membrane receptor binding are not as straightforward as in the case of intracellular receptors. The final actions or effect on a target cell will vary depending on the specific ligand and the type of target cell. But in all cases, the ligand, which is extracellular, is converted to an intracellular signal. This intracellular signal is called a second messenger. The most common second messengers are ions such as sodium, calcium, or cyclic AMP. What happens next varies a lot by cell type, and in the next few slides I'll examine how second messengers are generated. In one type of cell membrane receptor binding to its ligand, the receptor is also an ion channel. It allows a specific ion to pass across the membrane. This is shown in the box labeled number 1. When a ligand binds to the receptor, which is also a channel, opens, and the ion is allowed to diffuse in a direction consistent with its concentration gradient. We call these cell membrane receptors chemically gated ion channels. In the second type, labeled number 2 on the screen, a G protein is linked to a cell surface receptor. This G protein is a separate membrane protein and activated when the ligand binds to the receptor. Once the G protein is activated, it will have an effect on other nearby proteins. In this slide, the other nearby proteins are an ion channel and an enzyme. The effect of binding to a G protein linked receptor will become a bit more tangible to you if we look at some very specific examples. Before we look at specific examples of the G protein linked receptor, I'd like to show you one for a chemically gated ion channel receptor. In this case, the ligand is acetylcholine, a neurotransmitter released by neurons that target skeletal muscle cells. Skeletal muscle cells have an acetylcholine receptor that is a sodium channel. When acetylcholine binds to the receptor, sodium moves into the cell. Thereby, the muscle cell becomes positively charged, and that will initiate contraction. You will learn more about this in skeletal muscle physiology section of the course. Now for some specific examples of G protein linked receptors, I'd like to go over how an intracellular message is generated. So G protein linked receptors are linked to a G protein that facilitates the production of this intracellular signal. The intracellular signal is a second messenger which causes the cell's response. The most common second messengers are cyclic AMP and calcium. Epinephrine works through a G protein linked receptor. When epinephrine binds to the G protein linked receptor shown in purple, the G protein is activated. The G protein then activates a nearby protein called adenylyl cyclase. Adenylyl cyclase is an enzyme that converts ATP into cyclic AMP. Cyclic AMP then activates other enzymes and proteins in the cell. In the case of skeletal muscle cells being a target, the skeletal muscle cells will increase in size when epinephrine binds to the G protein linked receptor. One of the most important features of cell signaling that uses a G protein is that a small extracellular signal, meaning not very many signals, can generate a very large response by creating intracellular signaling molecules. This feature is referred to as signal amplification. At step 1 on the diagram, the ligand binds to the receptor. In step 2, you see that as long as the ligand is bound, it will activate many G proteins. This amplifies the signal right away. At step 3, we see that G protein activates a nearby membrane protein, and step 4 shows the result that as long as the G protein activates a neighboring enzyme, multiple compounds are produced. Again, the signal is amplified. Each second messenger that is made through amplification continues to activate multiple enzymes. thereby amplifying the original signaling molecule, which was a single ligand. So in summary, cell membrane receptor signaling involves an extracellular ligand that is converted to an intracellular signal called a second messenger. The external ligand or signal is amplified to a stronger internal signal. This happens by a G protein being activated as long as a ligand is bound to the receptor, and multiple downstream components are produced at many steps. The cellular responses will be very diverse. Some examples include metabolic reactions like the synthesis of glycogen, maybe secretion of a substance, or even cellular growth. This ends the presentation on cell signaling. Thank you for listening.